And we have also another procedure that is during the evaluation. I mean, if an evaluator realizes once he has signed the legal document declaring its not involvement in those projects, but if he realizes once he received the expression of interest to be evaluated that there is a conflict of interest with one of them, for instance, sometimes this happened, there is the possibility for them to declare this and to be, not to continue the evaluation anymore. And also during the panel meeting in Brussels, they will be, they will not participate to the evaluation of discussion about that expression of interest. But very likely, I mean, generally those, if we had very few cases of this kind, and they were, very light, let's say, conflict of interest. Of course, they were not involved in the projects, but sometimes they found a very good friend involved or being the coordinator of a project, and so in that case, they declared a conflict of interest.
There are many drugs of interest, many questions addressed, so it's difficult to cover everything. What is the good equilibrium between covering large or deep?
Yeah, good, very good question. We also discussed this, of course, internally. I mean, what we would like to have is at least, let's say, one representative of each class, meaning a monoclonal, for example, and a cytokine or something like that. You're not just monoclonals and cytokines. And I mean, the overall number we were thinking of around, what would be a good number, would be around 5 to 6. I mean, it could even be a little bit more. I mean, we discussed about that, for example, if we would have 20 different compounds, I think it's not handleable anymore. I mean, of course, the bigger the size or the number of the compounds would be, the more statistical power would be behind them. Not a statistician, but that goes without saying. But I think we need to be realistic, honestly. I mean, we cannot handle 50 different compounds. It should also be compounds that, what I said, I mean, where a lot of immunogenicity data is already available. So it does not make sense if we would choose something which is in development, for example, because there might not be enough immunogenicity data available. And in addition, I think no one would provide such compounds anyway. But I would say, 5, 6, 7 would be a good number. Of course, there is I mean, you're definitely right. I mean, there is a restriction if this is really, if the things that we will find out on that is really transferable to each and every protein, of course. But I think it's even better than we have right now because we really have a sort of, we concentrate on single products within the companies and also on some academia, so the data is simply not brought together. So everything that we are doing would definitely increase the information that we have right now.
But even for 6 or 7 drugs, it would be difficult to cover every single project.
I agree. I mean, probably that's why we said these first work packages that we are proposing, that the experts should sit together at the beginning, expert from both sides, to really define which data are necessary or which data they think are necessary to draw conclusions on either predictivity of the prediction tools, on the other hand, clinical impact. And probably they also should define how many products make sense. I mean, if they come out and say, yeah, we need to have more than 5, okay, then we should have more than 5. But of course, it also needs to be handleable, I mean, in the project, because if we would take 50 or something, or 20, I think it's not doable. But you're right, I mean, this is one of the things we are aware of, that, of course, the statistical power is restricted a little bit. On the other hand, we need to make it feasible.
In the microphone now, please.
Yes.
Sorry. Yeah, go ahead.
Could you explain a little bit more over here?
Sorry.
Could you explain a little bit more about the roadmap, about how to get to this proposal? Is there also a competition among proposals of the companies?
How we got to that proposal?
Yes.
Yeah, that's a good question. Actually, I mean, companies already tried to file sort of this during the first IMI call. And we also formed a full consortium, but at the end it did not succeed. And so we already knew some companies that were interested in immunogenicity prediction. And in principle, you also know the companies. It's like in the academic field. I mean, I also know my counterparts in other companies. And in principle, when we knew that there is a new IMI call to be published, we simply, rang up each other and asked if the other companies would still be interested in doing that. And also in the meantime, after the first IMI call, we were in intensive contact and tried to approve on the things that were criticized in the rejection of our first attempt for the immunogenicity. And so we were already somehow prepared on these kind of things. And then, in principle, simply people came together and agreed on writing a proposal. And
but finally, there was only one proposal.
Yeah, there was for that subject. But I think, probably you can tell more. I mean, it was not so clear that immunogenicity is one of the subjects, right? I mean, that's what I understood during the process, right? I mean,
the immunogenicity?
No, it was not clear that we will be taken as one of the calls, right? I mean, there were probably different topics that were proposed by the EFPIA members, right?
No, by the EFPIA members, but there are, of course, several proposals presented for that topic.
Yeah.
And the EFPIA, I mean, in terms of immunogenicity, of course, that was the only one.
But I think it all was in the safety package, right? Safety.
Yes, in the safety package.
Right.
Yeah.
So there were packages, and the EFPIA or companies could submit proposals. So
in the same package, you had different other proposals or possible proposals? Or
yes, we have 4 pillars in IMI. We have the safety pillar, efficacy, and knowledge management, and the pillar of education and training. So we have 4 pillars, and within each of them, we can have either one or more than one topic presented.
Can I, sorry to interrupt, but my comment was following what Dominique just said. Let's say, of course, you have to limit the number of targets, otherwise it's... But let's suppose in the first 2 years you find a general rule that will open new avenues and so on. So the question is more for you, is more for Teresa and Michel. Do you have the flexibility in these tools to readdress in such a way the project? Let's say now we have a new rules, we can look maybe at 20 and to see whether it's different, or you're stuck.
To readdress which stage?
After 2 years, you rewrite the annex and say, now we know that to induce the antibodies IgG2 and so on, so we want to change it. Is it possible to?
Well, in theory, yes, because after 2 years, we will perform an interim review with independent evaluators, and at that point, yes, the results reached so up to that point, and the objectives of the project could be slightly, modified according to the results obtained and the judgment of the evaluators that may reorient, advise the reorientation of the project. So I don't know if Michel has.
No, I agree. There is clearly flexibility. So which means that if all partners agree at some point that the technical annex should be amended in a way or another, but there must be obviously an agreement within the consortium. They just submit their proposal to us, and we can indeed review this, and this can be done really rapidly. And if the argumentation is good, there is no reason why we should not accept it. We are there as facilitators. We don't want to be hurdles. So I think that if the interest of the project, all partners agree that there should be some changes, they propose the changes and they explain how they will implement it. And obviously, we should, if it's reasonable, there is no way there is no reason why we should not find a way to implement it. Knowing that what is fixed is obviously the budget, but this is something that was obvious for everyone, I'm sure.
Since I have the microphone, can I go ahead?
Yeah, go ahead.
I think there is another point that is specific for this topic, but then we consider in general. So how the data that are obtained are open to the community and so on, because in this specific case, antibodies against these biologics can be used in sports and so on to address, to see abuse of these drugs, like erythropoietin, growth hormone, and maybe you come up with a new idea. So are these things open also to, can be published during the period? Are these things?
Yes.
As far as I understood it, it has to be published at the end, right? This is what I understood from the IMI rules. Anyway, it has to be published at least at the end. I mean, I don't know if interim publications are doable.
Whenever you have publications, the dissemination can be done in any period of the project.
Okay.
Yes.
Okay.
Other questions, either on the IMI rules or guidelines, or workflow, or on the immunogenicity topic? No, I think that. There is another one.
Thank you, Magda Rosemüller, Yes, Business School. I had a question to what you explained earlier, how this project actually became into being. On the number of pharmaceuticals companies involved here, I would say there may be more who might be interested in the outcome of this, or ask in a different way, that patient data you find in different clinical settings might be related to products which come from other pharmaceutical companies. How will you manage this? And is there a way of they might be involved at a later stage? I don't know. I'm just thinking of how if this is really something to be contributing innovatively, it should be generally
right. I mean, honestly, if you think about the companies currently joining, not all the compounds that we were proposing, which is anyway just a proposal, are from the company. So there's not a compound from each company in the proposal, which is anyway just a proposal. So this is the one thing. So it's not just we choosing these compounds because they're coming from the companies who were interested. We were really choosing these compounds because there's a lot of data available and a lot of public research done on these compounds. So this is the one thing. The other thing is, and this is probably more a question for you, but I can tell you what I understood under that. As far as I understood, other companies could still join until the final proposal is, the full project proposal is signed, right?
Actually, they can join, yes, and during the negotiation of the project. So once the expression of interest has been approved, and then and after also the second stage when the full project proposal has been written, so there are already involved all the companies there, then additional companies can be approved, provided that all the consortium accepts it, of course. So it's during the negotiation of the full project proposal.
And we also think, I think that's, what we're trying to find is really general principles. So it's not just for these compounds. So the idea is really to try to find common principles. For example, on the prediction tools, which would also help companies which are not dealing with the compounds that we proposed here.
Information in companies which are not participating companies, which kind of is left out, it could be interesting to contribute. That is the idea.
I think the problem, honestly, will be, like I said at the beginning, we had the discussion for the first call. It's very hard to disclose these data from a company point of view. Therefore, I think it's quite unlikely, although I would not say it's impossible, but I think it will be quite unlikely that a company would join which would bring in a lot of data on their own compounds or something. That's why we would really like to rely on the things which are in the public domain, because then we are really independent upon the companies. So honestly, we propose here interferon beta, and Magistris is making Rebif, which is one of these compounds. So definitely my company would not disclose any of these informations, but we would be okay if we would use data for interferon beta because it's just very immunogenic and there's a lot of publications out and a lot of research in the public domain if it's coming from a public domain kind of thing, and that we can learn in general how predictive the predictive tools are, for example. But I would say I think it's unlikely that a company would join by providing really a lot of own data. So either they really would join because they can contribute something to the things that we're saying, for example, their own prediction tools, or assessing immunogenicity with their own assays or something, but I would say it's unlikely that a company would join just providing data, from my experience. But, I might be wrong, but from my experience, I think it's unlikely.
Other comments or questions? No? So I think we can close the session.